Hi everyone. In this video I'm going to go over experiment 7, Tim and I are going to go over experiment 7, spectrophotometric determination of acid dissociation constant, pKa, of bromocresol green. So you're very familiar with what a pKa is, but you may not be familiar with how to calculate one using spectroscopy. And we're actually going to use two methods in this experiment to calculate the pKa of bromocresol green, which is an indicator, and we're going to use spectroscopy to do that. Down below in the description, you're going to find the usual outline with the usual color coding with what you need to have done before lab, before you leave lab, and then when you get out of lab. So please do make sure that you take a look at that. In fact, it might be a good idea to take a look at that before you watch this video, because in this video we may touch on some of the things in that outline. In fact, we will. So let's, I want to talk a little bit about background, and then I'm going to turn it over to Tim, and he's going to talk about some of the actual things you have to do. Unfortunately, this is not a particularly straightforward experiment, and it has a lot of moving parts. The more familiar you are with all of these parts before you come to lab, the better off you're going to be. So by way of theory, let's talk about what we're doing here. So essentially what we have is we have HIN, or protonated indicator. The H meaning the proton is attached. This, in your case, is bromocresol green. And basically you have a proton attached to this molecule. Okay, it's actually this proton that's leaving. And when you do have the proton attached, this has a yellow color, and its lambda max is 470 nanometers, which is, blue light. So this appears yellow. When we add base and we raise the pH, then it turns into a blue color because it loses this proton. And this blue color has a lambda max of 635 nanometers, which is an orangish color. Okay, so this is basically what's going on here. Now, why is it called bromocresol green? Because at intermediate pHs, it's going to be a mixture of yellow and blue. And as we know, a mixture of yellow and blue is green. So what are we going to do here? Well, we're going to look at the Ka of this, which is products over reactants. So we have IN minus, the deprotonated indicator, and H plus as the products, and the reactant is the protonated indicator, which we call HIN, so we don't have to write H bromocresol green every time or whatever. This is the Ka expression. Okay, all the stoichiometry here is 1 to 1, so these are all raised to the 1 power, or there are no exponents. We can then plug this same thing into the Henderson-Hasselbalch, and in the lab manual we derive the Henderson-Hasselbalch for you, but essentially you just take the log of everything, okay, and then we get pH equals pKa plus log of this ratio of these two indicators. And in the past, this is essentially what you would plug into, and in fact you will plug into this when you make your buffer solutions, but in this case, when we're doing this by absorbance, it gets a little bit more tricky, because we have to look at the different absorbances, and the different molecules have different molar absorptivity coefficients, which means we can't assume that the absorbance of this is 0.5 and the absorbance of this is 0.5, that they both have the same concentration, because they don't have the same molar absorptivity coefficient. So essentially what we do is math in order to take care of this. The first thing that we need to do is realize that the concentration of the protonated form of the indicator and the deprotonated form of the indicator is a constant. So that is very important. Then we're plugging in here into the Beer's law, where basically the absorbance of A at pH 1, where all of the indicator is HIN because it's in a strong acid, equals EBC. And for the second one, IN minus, it's at pH 13. Why? Because it's all deprotonated at a high pH when there's a bunch of base in there. So here, A equals E, BC again. So this is basically what we're looking at. Now, what we're doing is we're plugging these values essentially into the Henderson-Hasselbalch equation. And how we do that math is not super important. It is derived for you in the lab manual, but what you want to realize is what this relationship means. Okay, so IN minus, concentration of IN minus, is at the top of the Henderson-Hasselbalch equation. But instead of just using the absorbance at any given pH, okay, the pH here, which we're going to make buffer solutions for, instead of just using the absorbance of this blue species, we use the ratio of the absorbance of the blue species to the absorbance when it's only blue species. At the bottom, we use the absorbance of the yellow species at whatever pH it is versus the absorbance when it's all yellow. And we can do this because the total concentrations are constant. So this is basically what we're looking at. So we can't just plug in the absorbance of B over the absorbance of A, or said another way, the absorbance of IN minus over the absorbance of HIN. We have to plug in this ratio, which makes the math more complex, but we're going to use Excel, so it's not really a big deal. So one way that we're going to solve for the pH, the pKa of this indicator, bromocresol green, is to essentially plug in the pH, plug in this relationship, and solve for the pKa. And then you're going to do that 5 times. There's another way we can do that. We could think about this relationship as a line, where y equals mx plus b. So I slightly rearranged this. I put the log of this relationship first plus pKa second, so it's mx plus b. Notice that m, the slope here, is 1. Why is the slope here 1? Well, there's no m term here, first of all. And second of all, because the pKa is a constant. So however much we change this relationship is how much we're going to change the pH. The magnitude is going to be the same, so therefore, basically m is 1. The slope is 1. And if we plot several different pHs, and this ratio of the log of the concentration of IN minus over HIN, which is this expression right here, then we do a linear regression. The B term is the pKa. So we're going to determine the pKa by solving this equation for pKa, and we're going to determine the pKa by making this graph and finding B, or the y-intercept, because we're looking at this as the equation of a line. Now, in order to do all of this, we're going to show you, Tim's going to show you how to do all of this in the video, but we want you to know what you need to have. Now, you have made solutions lots of times, so we are not going to spend a lot of time talking about making solutions, although we will do one example with the buffer once you already have the 1 molar acetic acid and the 1 molar sodium acetate. But you need 4.5 times 10 to the minus 4 bromocresol green. You need 100 milliliters of that, and you're going to use 1 milliliter of that in each sample that you measure the absorbance of. You need 0.1 molar NaOH from solid. We suggest you start from 6 molar solid and dilute. And you need 1 molar HCl from 12.5 molar. Make sure you're adding the acid to the water and not the water to the acid. Please have these figured out before you come. We are not going to go over this further in this video. Additionally, to make your acetate buffers, right, we're going to make buffers at different pHs, and those pHs will be assigned to you, and we're going to show you how to do this in a minute. You're going to need to start by making 1 molar acetic acid from 17 molar. You need 100 milliliters of that, and again, add your acid to water, not your water to your acid. Note, we're not going to go over how to make this solution, so make sure you have the C1V1, C2V2 before you get to class. All right, and then you need 1 molar sodium acetate. Sodium acetate's a solid, so you're going to have to look up the molar mass there, and you want to make 100 milliliters of that total. Once you have these 1 molar solutions, you're going to use the stock acetic acid acetate solutions to make 0.1 molar, there'll be more sig figs here of course, molar buffers at 5 different pHs assigned by your TA. In order to do that, you need to be familiar with the fact that the pKa of acetic acid is 4.75. So now I'm going to turn it over to Tim, who's actually going to start by going over how you can turn these 1 molar acetic acid and sodium acetate solutions into the 0.1 molar buffers. Again, for the highlighted solutions here, you need to figure out how you make these before you come to lab. There's a lot of moving parts here. The more work you do, the more prepared you are, the better off you're going to be.
So earlier in the video, Colin said that I would show you in Excel how to do your calculations to make your acetate buffers. However, we've realized that once you figure out how to do one of those solutions in Excel, you can essentially extrapolate it to do all of your solutions in Excel without having to do any extra work. So in order to help you spend more time thinking about how to do the experiment and hopefully learn, obtain a better grasp of how to understand the math that I'm about to drive for you, we're going to show you to do it on paper and leave figuring out how to get it into Excel up to you. So what we need to do is we need to figure out how to solve for the concentration of A minus and the concentration of HA in our buffer. So the first important thing to know is that since we want a 0.1 molar solution, we always know that we want the total concentration to be 0.1. So we have that to start with. So what we need to do now is we need to solve for these concentrations. So to do that, we're going to start with the Henderson-Hasselbalch equation for this experiment, which is the pH is equal to pKa plus the log of A minus over HA. So what we want to do is we want to solve for either A minus or HA. I'm going to choose to solve for A minus because that's what I decided to solve for. So the first thing we need to do is we need to isolate A minus in this equation. So in order to do that, we're going to subtract the pKa from both sides, and we're going to end up with pH minus pKa is equal to the log of A minus divided by HA. Now, in order to get rid of this log function that's currently attached to our A minus, we need to raise both sides of this equation, take 10 and raise it to the power of each side of the equation. So practically speaking, what that's going to look like is we're going to raise the left side of the equation. We're going to take 10 to that power, and we're going to do that to the right side of the equation as well, but 10 to the log of a number, or in this case, ratio of variables, is just that number, or in this case, the ratio of the variables. So it's going to be equal to A minus over HA. Now at this point, you've probably realized there's no way to get rid of HA by simple math tricks. So what I'm going to do is I'm going to use this second equation as a system of equations in order to solve for a two-variable problem. Because anytime you want to solve an equation that has two variables, you need two equations. Luckily, we have another equation conveniently up here for us. So the concentration of HA is going to be equal to 0.1 minus the concentration of A minus. It's important to solve for HA because we're going to substitute in for HA so that the only variable in our equation is the A minus. So now what we're going to go to is we're going to have 10 to the pH minus pKa is equal to the concentration of A minus divided by 0.1 minus the concentration of A minus. So from this point, all we need to do is some various tricky math things in order to switch this all around. So what we need to do first is we need to multiply both sides of the equation by 0.1 minus A minus. So we have 0.1 times 10 to the pH minus the pKa when we multiply both sides by that value. So we're essentially doing this, which will cancel out on this side. And then we're going to multiply it by on this side as well. But when I do it on that side, what I'm going to do is I'm going to distribute it to each of these two values. So the 10 to the pH minus pK is going to multiply by the 0.1, which is 0.1 times 10 to the pH minus pKa. And then it's going to be subtracted minus, because we're doing minus A minus times 10 to the pH minus pKa. And that all is equal to the concentration of A minus. So now we need to further isolate our A minus. So to do that, what I'm going to do is I'm going to add this value to the other side of the equation. So if I have 0.1 times 10 to the pH minus pKa, it's going to be equal to the concentration of A minus plus this value, which is the concentration of A minus times 10 to the pH minus the pKa. So now what I need to do is I need to pull this A minus out of this side of the equation. So my left side of the equation will remain the same, and my right side will be A minus times 1 plus 10 to the pH minus pKa. So what I did there was I just took the A minus out of this part, the A minus out of this part. So if I take A minus out of A minus, I'm left with 1. And if I take A minus times 10 to the pKa and I pull out the A minus, I'm left with 10 to the pH minus pKa. So now I have the concentration of A minus isolated. So I simply divide both sides of the equation by 1 plus 10 to the pH minus pKa, which will cancel out on this side of the equation. And on the other side, when I divide it, I'll be left with the concentration of A minus is equal to 0.1 times 10 to the pH minus pKa divided by 1 plus 10 to the pH minus pKa. And this whole thing will let me solve for A minus. So, now that I can solve for the concentration of A minus, I also need to solve for the concentration of HA. So the concentration of HA, if you look up top, the concentration of HA we said was 0.1 minus that value. So the concentration of HA is equal to 0.1 minus the concentration of A minus, which is 0.1 minus 0.1 times 10 to the pH minus pKa divided by 1 plus 10 to the pH minus the pKa, which is some nice simple math for you to be able to solve. So now you can have the concentration of the A minus, the concentration of the HA. So now what you need to do is you need to figure out how much of each of your solutions to use to make the appropriate concentration of A minus and the appropriate concentration of HA. Well, just like always, anytime you're trying to solve for a concentration given, or a volume given an initial and a final concentration, you can use C1V1 equal to C2V2. So in this case, your C2 will be your concentration of A minus or HA. Your C1 will be your initial concentrations, which are both 1 molar. Your V2 in all solutions, as it says in the manual, is 25 milliliters total. And your V1 is the variable that you'll be solving for. So it's important to remember that your total volume will be 25 milliliters. So you'll have some amount of acetic acid, some amount of sodium acetate, and then the most important part is don't forget, please don't forget, your 1 milliliter of bromocresol green. It's really important that you add the 1 milliliter of bromocresol green before you fill the volumetric flask to 25 milliliters. Otherwise, you won't have an appropriately 25 milliliter solution. So you'll add your appropriate amount of sodium acetate solution. You'll add your appropriate amount of acetic acid that you solve from the dilution equation. Then you'll add 1 milliliter of bromocresol green, and finally you'll dilute to 25 milliliters with water. So that's an example of one of the types of the buffers that you'll be making. You can substitute in whatever pHs you need to solve this for the various numbers, because if you look at this equation, the pH and the pKa are your two variables. Your pH will be whatever 5 pHs are assigned to you by your TA, and the pKa will be a constant. It'll be 4.75 because you're using acetic acid. So when you sub in the 5 different pHs assigned to you by your TA, you'll get your 5 different concentrations of sodium acetate and acetic acid you need to make solutions of those concentrations. So now that we've found the pHs of our various solutions, we can actually measure their absorbance and determine the pKa through both the Henderson-Hasselbalch equation and through the graph like we discussed, or like Colin discussed at the beginning of this experiment. So on this sheet, what I have is the various pHs that I measured at. You'll see I did more than 5, and I also have an absorbance at 410 nanometers and an absorbance at 540 nanometers. These are not the same numbers or the same wavelengths or the same pHs that you're going to be using in class, so please don't take these as the only possible options. They're just the ones that I happen to do in this example. So I put my indicator in acidic solution and found my absorbance at 410 and 540, and I put my indicator in basic solution and did the same thing. This way I can subtract out the low values, the essential 0 value of the 410 and the 0 value of the 540 from my measurements, as these are essentially blanks, because when it's in the pH 13 form, there is no HIN present in the solution. So if there's no HIN present in the solution, this is effectively our blank value. And the same goes for the IN minus in the acidic solution. So I measured my absorbances using my microlab, and now I need to correct those absorbances. So you'll see that I have, for each value, taken my absorbance and subtracted out the blank value that I just discussed. For each of the 410s, I took the absorbance and I subtracted out the IN minus absorbance at the acidic concentration, so that I had a corrected absorbance of my IN. In this case, I took my 0.026 minus 0.007, and my corrected absorbance is 0.019. You'll see that I dragged down, and I did the same thing for each of these, taking my absorbance and subtracting out that value. I did the same thing for the HINs, subtracting out the absorbance of the HIN in the wavelength at the basic solution, and I did the same thing where I dragged down and I subtracted it from each of my values. So now I have my absorbance of my IN minus and I have the absorbance of my HIN. So now what we need to do is we need to sub into the equation that Colin solved at the beginning, which I'm going to pull up briefly to show you one more time. So I now need to sub into this equation where I'm going to solve for the pKa eventually, and I'm going to make this same graph. So what I need to do is I need to sub this value into that equation. So if you look in the lab manual, it interchangeably uses this and IN minus over HIN. So if I go to my Excel sheet, you'll see that I have log of IN minus over HIN. And here I've solved for it. Now I'm going to walk you through how I solved for it in Excel. So what you need to do is you need to take your absorbance at your measured value, so of the IN minus, which in this case I have already corrected for my blank, so I don't need to do anything to that. And then you need to divide by the absorbance at the solution where it's fully IN minus. And the reason you're doing that is to get the ratio of the absorbances to figure out the effective concentration, because you can't just use the ratio of the absorbances because the molar absorptivity coefficients are different. So to do that, I took the IN minus absorbance and I subtracted out its blank value for the acidic solution. So I have the the absorbance of IN minus when the solution is fully in IN minus form, and I've subtracted out the blank value. So I took the B12 minus B11. So that solves for the numerator of this equation. It gives me the absorbance at wavelength B, in this case the absorbance of IN minus, divided by the absorbance of IN minus at pH 13, which I corrected for by subtracting out the blank value. Now in the denominator I did the exact same thing. I took the absorbance of the HIN and I divided that by the absorbance of HIN corrected for the blank value as well. I took the HIN absorbance when the solution's fully in HIN form at pH 1, and I subtracted out the absorbance from that wavelength at pH 13 when there should be no HIN present in the solution. So I have C11 minus C12. So that solves for that entire equation. I have the log of my absorbance of B divided by my absorbance of B at pH 13, and I have that full value divided by the absorbance of my HIN divided by the absorbance of HIN at pH 1. When I take the log of that whole thing, I've also chosen here to round to 2 digits because this number has many digits in it, so I've chosen to round it to 2 because that's usually the number of digits we report pKa's to, and I've solved for this portion of my equation. So I have the log of IN minus divided by HIN. So When I did that for one of them, all I did was drag down so that it used my absorbance values at each of my measurements in order to solve for that value. Now what I've done in this graph is I've graphed on the y-axis my pHs taken from over here, and on the x-axis I've graphed my log of IN minus divided by HIN concentrations, which are these values that I solved for here. And as Colin covered at the beginning of the video, when you do that, your equation is in the form y equals mx plus b, and our b value is our pKa. So if we look at our graph, we see that our b value, once you've added a trend line, in this case is 5.94. So according to the graph, the pKa of the material I used for this example is 5.94. Now we can check our value by actually solving the Henderson-Hasselbalch equation for pKa. So in this case, what I did is, if we look at the Henderson-Hasselbalch equation, it is equal to the pH is equal to, sorry, it's up here, pH is equal to pKa plus the log of that whole value. Well, I have my pH and I have my log of that whole value, so if I subtract the log of that whole value from both sides, I'll have pH minus this is equal to pKa. So in my Excel file, you can see that all I did was my pH minus the log value that I just calculated. And when I do that and drag it down for each of my solutions, you'll see that I have pKa's around 5.94. Of course, it's not exactly 5.94 each time because various errors I may have made while performing the experiment, but you can see that it is around 5.94 or even exactly 5.94 for some of the solutions in all these cases. So that is kind of a brief overview of what you're going to do in the lab. If you need help working through this specifically, you can find an appendix in the back of your lab manual. I believe it's appendix number 4, but don't quote me on that, where it goes through how to set up your Excel sheet, including how to set up these equations and how to set up the graph and add your trend line and all of that. However, there is an error in one of the formulas used in your lab manual, so I have rewritten that appendix and I will post it on Blackboard for you to use for this semester. So you'll have a copy of the corrected appendix on Excel so that you can use that rather than the version in your manual. So that's everything you're going to need for this week's experiment. Like Colin said before, it's really important that you're prepared for this experiment. There's a lot of math involved in this experiment, and if you spend the entire lab period trying to figure out all the math and figure out what you're going to be doing with no basis to start on, there's no way you're going to be able to finish this experiment in time. So it's really important that you are prepared for this experiment before you come to lab. We strongly recommend that you fill out as much of your buffer calculations and as much of your graphing calculations as you can before you come to lab. You can theoretically set up your calculations to make all of your solutions that Colin covered earlier. You could set that up ahead of time, and you could even set up your Excel sheet to be prepared to solve for the volumes of your sodium acetate and acetic acid solutions to make your buffer solutions as soon as your TA gives you the pHs. You could set up your Excel sheet using the equation that I showed you before, so that all you need to do is type in your pHs and your volumes of solutions will fill themselves out automatically. And I strongly recommend that you consider doing that prior to the lab so that you don't have to spend your precious time in lab solving for those equations. So as a kind of reward for sticking around so long through this math-heavy, complicated video, I've added to the screen an example of one of the potential solutions you could be instructed to make by your TA. At a pH of 3.6, I've used the equation I showed you before to solve for our volumes in moles. So we need 0.00234 1.4 or so moles of acetic acid and 1.61 times 10 to the minus 4 moles of sodium acetate, which means that in a 25 milliliter volumetric flask, I need 2.34 milliliters of the 1 molar acetic acid and 0.162 milliliters of the 1 molar sodium acetate. And then I used the Henderson-Hasselbalch equation, which I haven't shown you here, but I used that to calculate and make sure that the pH would be the same given the known pKa of acetic acid. So that's kind of your reward for having stuck around. You can kind of get an idea of what you should set up when you perform this yourself. And make sure that you include this last column here, which uses the volumes and converts them to concentrations in order to sub in the Henderson-Hasselbalch equation to double check your numbers and make sure it works, because this pH should be equal to this pH. We use this one to solve for these moles and these volumes, and then we use those volumes to solve for the pH using the Henderson-Hasselbalch equation, just to double check our work. So, thank you for watching the video this week, and again, we urge you to be very prepared, and good luck in your lab this week.